Get started. So, we talked about gas chromatography last time. Let's talk about liquid chromatography. The liquid chromatography is a technique where the mobile phase is a liquid. It's a much older technique than gas chromatography, but it was overshadowed by the development of gas chromatography in the 50s and 60s. And it's coming back it's currently the dominant type of chromatography. It's even replacing GC in its more traditional applications because of the development of more advanced systems, the high performance liquid chromatography. So liquid chromatography can be applied to the separation of any compound that's soluble in a liquid phase. It's more useful in the separation of biological compounds, synthetic or natural polymers, or inorganic compounds, stuff that doesn't vaporize as easily. And the liquid mobile phase allows it to be used at lower temperatures that are required by GC. So it's much better for separating compounds that are less thermally stable, so more likely to be destroyed by the higher temperatures in GC. The retention of solutes in liquid chromatography depend on their interaction with both the mobile phase and the stationary phase, while gas chromatography retention is based on volatility and interaction with the stationary phase. Liquid chromatography is more flexible in optimizing separations. You can change either the stationary or the mobile phase. Most liquid chromatography detectors are non-destructive, while most GC detectors are destructive. Liquid chromatography is better suited for preparative or process scale separations, when you're trying to separate out material for further steps in synthesis, for example. Some disadvantages: it has greater peak or band broadening. There's much higher diffusion coefficients of solutes in gases versus liquids. But basically, there's a bigger effect of peak broadening or band broadening. And there's low and high performance liquid chromatography. So there's a lot of different types of liquid chromatography based on different stationary phase and mobile phase combinations. And each type can be further characterized based on its overall efficiency or performance. So you have organic soluble, so you hexane, MEOH, DHF, and water soluble, non-ionic, ionic. These are different molecular masses and different things. You can have normal phase absorption, reversed phase bonded, small molecule for gel permeation, reversed phase for bonded for non-ionic, paired ion and ion exchange, size exclusion or gel permeation in organic soluble, water soluble. You have size exclusion with gel filtration, and then ion exchange and reversed phase bonded for the bigger molecular mass stuff. So low performance liquid chromatography methods use a large non-rigid support material. The particles are greater than 40 micrometers in diameter. It has poor system efficiencies and large plane heights. You often have broad peaks, poor limits of detection, long separation times, and the columns can only tolerate low operating pressures. You can use gravity flow or peristaltic plump to apply the mobile phase to the column. The advantage of low performance liquid chromatography methods is that they are cheaper and simpler to set up. So if you're just trying to separate your starting materials for, get rid of unwanted impurities for some sort of synthesis reaction, much easier to set up a gravity-based low performance liquid chromatography method than some sort of high performance one. So for example, column chromatography, which is an example, you have just a column of material and built on top of a porous glass plate with a column head and a solvent reservoir above. and you fill it and let the solid, you apply the sample directly to the top of the column. And then the detection is by just collecting different fractions with data analysis of each fraction. So you click, regularly, you fill various beakers with various fractions of the column. And this is good for various reaction stuff. So the advantages, it's got simple system requirements, it's inexpensive, it's popular in sample purification. It's good for sample purification because you just need to get the right sort of section. It's used in the removal of interferences from samples. It's used in some analytical applications, but not commonly because of the low efficiency, long analysis time, and poor limits of detection. It is good for purification, not so good for analysis. It's also easier to do large quantities. High-performance liquid chromatography uses a small, uniform, rigid support material with particles less than 40 micrometers in diameter. They're usually 3 to 10 micrometers. It has good system efficiencies and small plate heights. You have narrow peaks, low limits of detection, shorter separation times in the columns, but the columns can only tolerate high operating pressures, and faster flow rates. The faster flow rates is good. The higher operating pressure is something you have to worry about just to work with. So a typical high performance liquid chromatography system is also a high pressure one. The higher operating pressures needed for mobile phase delivery require special pumps and other system components. So you have some solvent reservoir, a pump, an injector, a column, a detector, and an output. The sample is applied using a sole closed system, i.e. like an injection valve, where you could fill a fixed volume with your sample and put that into the line. Detection uses a flow-through detector of some sort. And a modern system will also have the ability to control and vary the solvent involved. So this is a solvent with a regulated helium source that can help you portion which pressurize the solvent reservoirs that feed into a proportioning valve. And then you have the pump and a pulse damper and drain valve and so on. So it has a fast analysis time. It's easy to automate. It has good limits of detection. It's the preferred choice for analytical applications. It's also popular for purification work. The biggest disadvantage is it's expensive, and the sample capacities are a lot lower. So if you're doing some sort of big purification job, you really don't want to do it with a high-performance liquid chromatography system, because you'll have to do it over and over and over again to separate things out. But it's good for analytical stuff work. So the retention and elution of solutes in liquid chromatography depends on the interaction of the solutes with both the mobile and the stationary phases. And to describe how well solutes are retained in the column with different solvents, the terms weak mobile phase and strong mobile phase are used. So you have a strong mobile phase. There's some solvent that quickly elutes solutes from the column, i.e. has a small K prime. And this occurs if the mobile phase is very similar to the stationary phases, intermolecular interactions with the solvents. So for example, a polar solvent would be a strong mobile phase for a column containing a polar stationary phase. And you get rapid elution in a few minutes for all compounds in the mixture, for example. The weak mobile phase is a solvent that slowly elutes solutes from the column, i.e. a high solute retention or a large K prime. This occurs if the mobile phase is very different from the stationary phase in its intermolecular solutes. So a non-polar solvent would be a weak mobile phase for a column containing a polar stationary phase. So the strong mobile phase is similar to the stationary phase. The weak mobile phase is different from the stationary phase. So this is a slow elution of around 20 minutes for all the compounds in the mixture. And whether a solvent is a weak or strong mobile phase depends on the stationary phase being used. So for example, hexane is a weak mobile phase on a polar stationary phase, but a strong mobile phase on a non-polar stationary phase. So it has to do with how it compares to the stationary phase. Similar to gas chromatography, solutes can be eluted from a column by either using a constant column conditions or a gradient dilution. An isocratic dilution uses a constant mobile phase composition to elute solutes. It's simple, it's inexpensive, but it's difficult to elute all solutes with good resolution in a reasonable amount of time. This is sort of the general elution problem we've discussed. So you can identify sort of a best composition to get optimal separation for constant here, which in this case looks like it would be somewhere around the 0.3 here is better than either 0.25 here or 0.25 for separating all of these. But you could also vary the concentration over the range. So similar to GC, solutes can be eluted from a column by using either constant column conditions or a gradient one. In the gradient one, you steadily change the concentration of the mixture. So generally you go from a weak mobile phase to a strong mobile phase. So here solvent A is the weak mobile phase, solvent B is the strong mobile phase, and the solvent change can of course be stepwise, linear, or nonlinear. But so general idea, you're going to go from weak to strong to basically steadily speed up the process and even out the process. So here's an example with a gradient mixture of 30 amino acids, and here's the gradient profile where you have this nonlinear process and then this linear process, and then going from weak to strong. And in choosing a mobile phase, you need to do several things. The type of stationary phase is used, which determines what will be strong or weak. The solubility of your solutes, the viscosity of the mobile phase, the type of detector that's used, and the solvent's background signal in that detector. The purity of the solvents, and the mixability of the solvents for a gradient dilution. They need to mix. So here's a selection of various mobile phases. These summarized properties. The refractivity index is important for the detector. The viscosity is important for flowing through. The boiling point is about what temperatures you can work in. The polarity index helps determine whether it's strong or weak, and the elute strength is Now, types of liquid chromatography are classified according to the method of solute separation. There's adsorption chromatography, where the solute is absorbed on the surface. Partition chromatography, where the solute gets dissolved in a liquid phase that is coated on the surface. Ion exchange chromatography, where mobile ions interact with an anion exchange resin. Affinity chromatography, where there's antigens or antibodies, something like antibodies bound to the surface, and the antigens interact with them. And then there's size exclusion chromatography, which generally, the porosity of the state of the structure affects how things flow. So you could have gel permeation chromatography, gel filtration chromatography, and gel chromatography. And they depend on whether, they slow down large molecules or slow down small molecules. We'll go into some detail. So absorption chromatography separates solutes based on their adsorption to underivatized solid particles. This is like gas solid chromatography, and the same materials used as the stationary phase and the support surface. So the solute gets absorbed on the surface. The advantage is it will retain and separate some compounds that can't be separated by other methods, like geometrical isomers. The disadvantage is you have very strong retention of some solutes. It can cause catalytic changes in the solutes, and the solid support may have a range of chemical and physical environments. You can have non-symmetrical peaks and variable retention times because of that. So you can have things like silica, which is the surface is slightly acidic. Its general purpose or basic compounds. Alumina, slightly basic, general purpose or acidic compounds. Charcoal has a nonpolar surface. It's good for nonpolar compounds. Fluorosol is strongly acidic. Its general purpose or basic compounds. Polyamides are basic. It's good for phenols and aromatic nitro compounds. And others, things like clay, chlorine, diatomaceous earth, cyanide, that are relatively nonpolar, which are good for polar compounds. For polar supports like silicon and alumina, the weak mobile phase is a nonpolar solvent, like hexane and benzene, etc. And the strong mobile phase is a polar solvent, like water or methanol. For nonpolar supports like charcoal, the weak molar phase is a polar solvent, and the strong mobile phase is a nonpolar solvent. There's some common applications for absorption, liquid chromatography, purification of synthetic organic compounds from reaction mixtures, separation of geometrical isomers like ortho, meta, para, cis, trans, etc. Partition chromatography separates solutes based on their partitioning between a liquid mobile phase and a liquid stationary phase that's coated on a solid support. The support material is usually silica, and this originally involved coating the support with some liquid stationary phase that wasn't readily soluble in the mobile phase. The two main types of partition chromatography based on the type of stationary phase is normal phase liquid chromatography and reversed phase liquid chromatography. Oh, sorry. The normal phase liquid chromatography is the partition chromatography where the stationary phase is polar. Normal phase liquid chromatography strongly retains polar compounds. The weak mobile phase is a non-polar liquid or an organic solvent. The strong mobile phase is a polar liquid like water or methanol. And the stationary phase has to have a low mixability with the mobile phase, so the stationary phase is not dissolved from the column. So some examples of liquid nonpolar stationary phase: dimethyl ethyl sulfide, ethylene glycol, water, ethylene diamine. And the liquid stationary phases will slowly bleed from the column, changing the properties and solute retention times. So commonly these days, people use stationary phases chemically attached to the support, like say anapropyl, aminopropyl, or propylethylamidine bound to the silicon. And that has the advantage of doesn't bleed away. So for stationary phase materials, in normal phase chromatography, the stationary phase is initially more polar than the mobile phase, and separation is driven by a difference in polarity. Stationary phase is silica or silica support derivatized with aminodiol or cyanofunctional groups, generally. So this is sort of a schematic of normal phase separation. And gray is more polar, blue is less polar. So the gray stuff is held back, the mobile phase is less polar, so partitions. And so the analyte will partition preferably into the stationary phase compared to those analyte molecules that are non-polar inclusions. The initial mobile phase is less polar. For example, 7030 CH30CN and methyl and oxide. So polar analytes are partitioned more heavily into the stationary phase. And then it transitions to MeOH and then to very polar H2O. And most polar species will come off with water. So this is sort of a three-phase. So 70-30 acetonitrile methanol, then 100% methanol, and then 100% water. So the common applications for nonpolar liquid, so normal phase liquid chromatography, sorry, is the purification of synthetic organic and inorganic compounds from reaction mixtures, and the general purpose separation of polar nonpolar compounds from the samples in a nonpolar solvent. So here's an example of automated chromatography purification. Reverse phase liquid chromatography is partition chromatography where the stationary phase is nonpolar. It's the reverse polarity of normal phase. It retains nonpolar compounds most strongly. The weak mobile phase is usually a polar liquid water. The strong mobile phase is a more nonpolar liquid methanol or acetonitrile. The stationary phase must have a low mixability, of course. So examples are of station with the mobile phase, so the stationary phase isn't dissolved from the column. So some examples are heptane, hydrocarbon polymers, squalene, dimethyl polysiloxane. So comparison, the stationary phase for reverse phase liquid chromatography is nonpolar. The weak mobile phase is a polar liquid. The strong mobile phase is a more nonpolar liquid. And then for normal phase liquid chromatography, the stationary phase is polar. The weak mobile phase is a nonpolar liquid. The strong mobile phase is a polar liquid. This is used, reverse phase chromatography is used to separate relatively nonpolar analytes. The mode of separation depends on differences in polarity. The typical stationary phase is C8 or C18 chains bonded to silica, or polystyrene diphenylbenzene. You start with a more polar mobile phase, and hydrophobic analytes will associate with hydrophobic stationary phase, and the mobile decays become more nonpolar, pulling hydrophobics off of the stationary phase. And so things like C18, C8, deoxydecyl, octyl, ethyl, cyclohexyl, or phenol are things that are put used. C18 and C8 are the most common. So bound stationary phase is more common than liquid phase. So C8 and C18 stationary phases function primarily through surface interactions or as very thin layers. So if the stationary phase becomes more 3D, broadening due to mass transfer resistance can result. So here, more polar things travel fast. So more nonpolar things travel more slowly. So some things that this is used for a postmortem blood sample. More polar species elute first, less polar species elute after. The mobile phase becomes sufficiently nonpolar. So this is an example of a blood sample. This is actually the most popular type of liquid chromatography because it separates a wide variety of nonpolar and polar solutes. And part of the reason it's popular is the weak mobile phase is a polar solvent, i.e. water. So it's great for stuff that is in aqueous-based samples, such as biological compounds. So normal phase is good if you have stuff in nonpolar solvents. This is good if you have stuff in polar solvents, i.e. aqueous. So normal phase chromatography, you have a low polarity mobile phase, medium polarity mobile phase. Here, reverse phase, you have a high polarity mobile phase and a medium polarity mobile phase, where A is more polar than B, which is more polar than C. So this is commonly used for the purification of biological and organic compounds present in aqueous solutions, pharmaceutical analysis, drug quantification and quality control, protein and peptide mapping, analysis of soil and water samples, and clinical analysis of blood and urine samples. This is great for anything in an aqueous solution, which are pretty dang common. Ion exchange chromatography separates solutes by their absorption onto a support containing fixed charges on its surface. So a high concentration of a competing ion is often added to the mobile phase to elute the analytes from the column. So the general idea is it separates things by their amount of ionization. You separate ionic species. The driving force is ionic charge density. The stationary phase is either negatively charged, a cation exchange, or positively charged, an anion exchange. The stationary phase can be weak or strong, so weak cation exchange, or weak anion exchange, or strong cation exchange, or strong anion exchange. And these are these ion exchange resonance. So anion exchange stationary phase will attract negatively charged ions. Cation exchange stationary phase will attract positively charged ions. They're silica beads with bonded ionic moieties. So cation exchangers have fixed negatively charged groups used to separate positively charged ions. Anion exchangers have fixed positively charged groups used to separate negatively charged ions. So here are some various exchange mediums. The cation exchanges are acids, the anion exchanges are bases. And the charge groups that make up the stationary phase can be placed on several different types of support materials. Cross-linked polystyrene resins are very common for the use with the separation of inorganic ions and small organic ions. Carbohydrate-based resins for low-performance separations of biological molecules like dextroaggregose cellulose, and silica-based supports for high-performance separation of biological molecules. They commonly use these ion exchange resins. And a strong mobile phase in IEC contains a high concentration of some competing ion for displacement of the sample ion from the stationary phase. So here are some examples of ions that can be used. Or some solvent that has a pH which decreases the ionization of the analyte or stationary phase. So factors that affect the mobile phase there are things like the mobile phase pH, especially for weak acid or base analytes and weak acid or base stationary phases. The mobile phase concentration of a competing ion and the type of competing ion. So here's like the net charge of a protein as a function of pH. And so you can switch that, whether it be attached to anion exchangers or cation exchanges as a function of pH. So initially, the stationary phase is associated with some weakly bound counterion. Analyte ions replace these counterions and are bound to the column for electrostatic interactions. And then as the experiment progresses, stronger counterions are introduced that displace the analyte ions, allowing them to elute. And here's an example. So common applications: removal or replacement of ionic compounds in samples. So pre-treatment of samples to get rid of ionic compounds. Separation of inorganic ions and organic ions. And the analysis and purification of charged biological compounds like amino acids, proteins, peptides, and nucleic acid. Affinity chromatography separates based on the use of immobilized biological molecules and related compounds as in the stationary phase. So you can use, this basically uses antibodies and antigens. So you can either use antigens to identify antibodies or antibodies to identify antigens. So this is based on the selective reversible interactions that characterize most biological systems, these sort of lock and key structures you get. So the binding of an enzyme with its substrate or a hormone with its receptor. You immobilize one of a pair of interacting molecules onto a solid support, and the immobilized molecule in the column is referred to as the affinity ligand. They're often used in biological applications. The analyte has a preference or affinity to bind to another substance, like the glucose binding protein has an affinity for glucose. These can be used to preferentially bind glucose for quantitative analysis. Other binding sp ligands include antigens, enzymes, biochemical receptors, things like that. You can have very high specificity ligands, which bind to only one of a very few closely related molecules, like antibodies, which bind to antigens, and antigens which bind to specific antibodies. You can have inhibitors or substrates, which retain enzymes, or nucleic acids, which bind to complementary nucleic acids. Or you can have more general or group-specific ligands, like lectins bind to glycoproteins, carbohydrates, and membrane proteins. Triazine dyes bind to NADH or NADPH-dependent enzymes. Phenylboric acid binds to cis-diol-containing compounds. Protein A and protein G bind to various antibodies. Metal chelates bind to metal-binding proteins and peptides. Doesn't necessarily have to have a biological origin. And due to the very selective nature of biological interactions, the solute of interest is often retained with very little interference from other compounds in the sample. So it's very good for purifying out a particular thing. And a weak mobile phase is usually a solvent that mimics the pH, ionic strength, and polarity of the solute and ligand in their natural binding environment. And a strong mobile phase is a solvent that produces low retention by either decreasing the binding constant or it displaces the solute by the addition of an agent which competes for solute sites on the column. Kind of thing. I think more common is decreasing the binding constant because you can change the pH or other things like that. And that will often cause things to not bind anymore. Or you can have some other thing which binds better and helps separate. So biospecific elution is where solutes are eluted by a mobile phase that contains a compound which competes with sample solutes for the ligand's active sites. This is very gentle. It's useful in the purification of active biological molecules. It produces a slow elution with broad solute peaks. Non-specific elution is where you change the conditions of the column to disrupt the interactions between sample solutes and a mobilized ligand. So you change pH or ionic strength. This is harsher than the biospecific elution. It gives narrow peaks and faster runtimes. It's commonly used in analytical applications. So it's used for the purification of enzymes, proteins, and peptides, the isolation of cells and viruses, the purification of nucleic acids, specific analysis of components in clinical and biological samples, the study of molecular interactions, things like that. So this is an example of purification of the histag protein using a pH change. Size exclusion chromatography separates molecules according to differences in their size. So molecular exclusion chromatography excludes large molecules from the porous structure of the support material. So large molecules pass through faster than smaller molecules. The SEC is basically uses a support material that has a certain range of pore sizes. And what will happen is that the smaller proteins will travel through these beads while the larger proteins will travel around them and elute more quickly. So as the solute travels through the support, small molecules can enter the pores while large molecules can't. So the larger molecules sample a smaller volume of the column. They elute before the smaller volume molecules, and the separation is based on size or molecular weight. It has to do with the different interactions of the solutes with the flowing mobile phase and the stagnant mobile phase. There's no true stationary phase. The stagnant mobile phase acts as the stationary phase, but it separates things on size. Gel permeation chromatography uses a hydrophobic stationary phase and an organic mobile phase. It's the SEC analog to reverse phase chromatography. Gel filtration uses a hydrophilic stationary phase and an aqueous mobile phase, the analog of normal phase. And the mode of separation is by molecular size. Molecules that fit into pores are retained. Molecules that are too large to fit are swept along. That's how size exclusion works. There are other methods that work the other way around, where smaller molecules travel faster than larger molecules, but they aren't exactly chromatography. It doesn't have weak and strong mobile phase because retention is based only on the size and shape of the analyte and the pore distribution of the support. For gel filtration, it's gel filtration chromatography if an aqueous mobile phase is used. It's gel permeation chromatography if an organic mobile phase is used. It's often used for separating biological molecules, like separating proteins from peptides, the separation and analysis of organic polymers, and molecular weight determination of various things. Because again, it separates things based on their size. So it can separate them based on their molecular weight. And it can be very useful for purification because let's say you have some large molecule you made and a bunch of the precursor material left behind that's much smaller. The large molecule will separate out first while the precursor stuff will take longer to travel through. So the largest analyte gets eluted from the stationary phase pores first. The smallest analyte penetrate the deepest to the stationary phase pores and elute last. Both size and shape matter. So linear molecules have a higher hydrodynamic volume. They consume more space than less linear ones. So both of these are C10H8, but this one is going to be bigger because it's more, it's longer. And here's a calibration curve of molecular weight versus elution time for an analytical gel filtration experiment. The next thing is detection. So here are some common types of liquid chromatography detectors. You have a refractive index detector, a conductivity detector, a UV visible absorptions detector, an electrochemical detector, and a fluorescence detector. And as in gas chromatography, the choice of detector will depend on the analyte and how the LC method is being used. Is it analytical or preparative? So a refractive index detector, it's got poor selectivity, relatively poor sensitivity, but can be used for any component that differs in refractive index from the elute can be detected. And despite its low sensitivity, but it can't be used to perform a gradient analysis because you can't change the mobile phase. UV visible has moderate selectivity and good sensitivity. This can detect a wide variety of substances that absorb light in the UV, so are visible from 190 to 900 nanometers, and the sensitivity depends strongly on the component. Fluorescence has good selectivity and excellent sensitivity. The components that emit fluorescence can be detected selectively with high sensitivity. This is often used for pre-column and post-column derivatization. Conductivity has moderate selectivity and good sensitivity. Ionized components are detected. The detector is used mainly for ion chromatography. Electrochemical has good selectivity and excellent sensitivity. Electric currents are detected that are generated by electric oxidation reduction in reactions. Electrically active components are detected with high sensitivity. So the ideal characteristic for a detector is that it's selective for the analyte of interest. It's insensitive to the mobile phase. It shows a linear response over a wide range of analyte concentrations, has a low limit of detection, high signal to noise. It's highly sensitive. It has a large slope on signal versus concentration plot, and it's stable over time. It doesn't drift. So a refractive index detector measures the overall ability of the mobile phase and its solutes to refract or bend light. It's one of the few universal detectors available for liquid chromatography, where if there's only the solvent, the light will be bent here, but if there's a sample present, it will move. It's non-destructive and a universal detector. It's applicable to the detection of any solute in liquid chromatography. It's applicable to preliminary liquid chromatography work where the nature and properties of the solute are unknown, provided that the concentration is high enough for detection. The disadvantages, it has a relatively high limit of detection. You need to have 10 to the minus 6 to 10 to the minus 5 molar. And it's very hard to use with a gradient solution. It's possible because you can use a reference side and a sample side and run them simultaneously, but it's hard. So light from a source passes through flow cells containing either the sample stream or a reference stream. And when the refractive index is the same between the cells, no bending of light occurs at the interface between the flow cells, and the maximum amount of light reaches the detector. And as the solute elutes, the refractive index changes between the reference and sample cells, and light gets bent as it passes through the flow cell interface, and the amount of light reaching the detector is decreased. So on one side, we have the sample, on the other side, we have the reference. Another common thing is a UV visible absorption detector, which measures the abilities of solutes to absorb light at some particular wavelengths in the ultraviolet or visible range. This is the most common type of liquid chromatography detector. There's three common types. You have fixed wavelength detectors, which only detect one wavelength. You have variable wavelength detectors, which can vary, which have a monochromator, so you can pick your wavelength in question. And then you have photodiode array detectors, where your monochromator, where you separate them out after pass through the sample onto a photodiode array and can detect several wavelengths simultaneously. The fixed wavelength detector absorbs only one given wavelength that's monitored by the system at all times. This is usually 254 nanometers. It's the simplest and cheapest of the UV visible detectors. It's limited in flexibility and limited in the type of compounds that can be monitored, but it's simple and inexpensive. A variable wavelength detector, where you only monitor one wavelength at any time, but any wavelength in a wide range can be selected. So the wavelengths will vary from 190 to 900 nanometers. This is more expensive. It requires more advanced optics, but it's also more versatile and can be used for a wider range of compounds. And a photodiode array detector operates by simultaneously monitoring the absorbance of solutes at several different wavelengths. This uses a series or array of several detector cells within the instrument, with each responding to absorbance at different wavelengths. And the entire spectrum of a compound can be taken in a minimum amount of time. This is useful in detecting the presence of poorly resolved peaks or peak contaminants. So you measure across a capillary tube. This yields poor results because B is very small. So you usually have something more like this, where you have fiber optic cable from the source, fiber optic cable from the detector, and then you run in this sort of Z-shaped shell, where the mobile phase passes along the path of the light here. And selectivity can be enhanced by choosing the proper wavelength. So this is how the photodiode array works. So you have a light source, you go through the flow cell, and then you have a diffraction grating in a photodiode array. And you can detect any compound that absorbs at the wavelength being monitored. So common wavelengths are 254 nanometers for unsaturated organic compounds, 260 nanometers for nucleic acids, 280 or 250 nanometers for proteins or peptides. And absorbance detectors can be used with gradient dilution, because the wavelength being monitored is above the cutoff range of the solvents. As long as the wavelength being monitored is above the cutoff range of the solvents being used in the mobile phase. And the limits of detection for fixed and variable UV visible absorption detectors are 10 to the minus 8 molar. For photodiode array detectors, it's more like 10 to the minus 7, but you get a whole spectrum. A fluorescence detector is a selective liquid chromatography detector that measures the ability of eluting solutes to fluoresce at a given set of excitation and emission wavelengths. So you excite it as you go through the flow cell, and you have emission, which you detect. Fluorescence can be used to detect any compound that absorbs and emits light at the chosen set of excitation and emission wavelengths. Relatively few compounds undergo fluorescence, which gives you high selectivity and a very low background signal, and very good limits of detection, 10 to the minus 10 molar. This is typically used for drugs, food additives, certain environmental pollutants, any compound that can be converted into a fluorescent derivative, like alcohols, amines, amino acids, and proteins, you can tag them. And it can be used with gradient dilution, but it does require extremely pure mobile phases because trace impurities can affect the background signal or quench the fluorescence. A conductivity detector is useful for ion exchange chromatography, detecting ion compounds. This uses a Wheatstone bridge. The detector measures the ability of the mobile phase to conduct a current when placed in a flow cell between two electrodes, and the current conducted within the cell will depend on a number of types of ions present in the mobile phase. So, R1 and R2 are just resistors. R3 and R4 are, the resistor is the sample cell. You're running a current through the sample, and you're comparing it to a reference flow. So two electrodes placed in the mobile phase each correspond to one arm of a Wheatstone bridge. Over here. And the impedance between the electrodes changes, presiding an out of balance signal here, which can be easily detected. It can be used to detect any compound that is ionic or even weakly ionic, has high selectivity, low background signal. Limits of detection are around 10 to the minus 6 molar. This is used for food and components, industrial samples, environmental samples. It can be used with gradient dilution because it just requires constant ionic strength and pH of the mobile phase, and that the background conductance of the mobile phase is sufficiently low. Electrochemical detector is used to monitor any compound in the mobile phase that can undergo an oxidation or reduction. This is electrochemical detection in liquid chromatography is referred to sometimes as LCEC. Generally, you have two or more electrodes which monitor the current that's produced by the oxidation or reduction of eluting compounds at some fixed potential. And generally, the electrical output is an electron flow generated by a reaction that takes place at the surface of the compounds. It can be used to detect any solute that may undergo oxidation or reduction. The detectors can be made specific for a given compound or class of compounds by properly choosing the condition of the electrodes. It has a high selectivity, a low background signal, but it only works for hidden solutes. The limits of detection are really great, 10 to the minus 11 molar. And this is due to the extreme accuracy with which chemical measurements, especially current measurements, like electrical measurements, especially current measurements, can be made. You can detect things that can be detected by reduction, or things like aldehydes, ketones, esters, unsaturated compounds. Things that can be detected by oxidation are phenols, mercaptans, aromatic amines, hydroxy compounds. So a simple liquid chromatographer setup has a solvent reservoir, some column with packing material, detection, and collection. Here's an example. Solvent, some sand, the stationary phase, glass wool, and collection and detection, and you're just collecting in beakers, partition collected. High performance liquid chromatography has multiple mobile phases with a mixer, a pump, a sample injection port, which may be hooked up to an auto sampler, a column, a detector, and collection, all tied to a computer. And it has a sophisticated detector, which is your optical, maybe mass spec. So here's sort of a block schematic. And here's an example of one. Sample organizer over here, various detector options, column management options, the sample manager, binary solvent. And the injector has some sample loop with a fixed volume that you can switch into the column to feed but you can feed it from the outside, or you can switch it in and feed it from the column and pump. So while in the load position, the column is going to bypass the sample loop volume. You fill it with your sample, and then switch it in the inject thing. So the mobile phase passes through the sample loop where it carries the sample along the column. So this is the injector. Because you're injecting into a very high pressure system, something like 300 atmospheres, it uses a manual or automated injector molecule. So in the load position, the mobile phase flows through the injector, bypassing the sample loop and directly into the column. During load, a syringe is used to fill a fixed volume loop with the sample. And when rotated to the inject position, the mobile phase flows through the loop, picking up the sample and bringing it onto the column. You need a very even delivery of mobile phase to the column. You use check valve. So this is a pump for a high pressure performance liquid chromatography pump. You need very even delivery of mobile phase to the column. You use ball check valves to prevent flow in reverse, and a sapphire piston that withstands high pressures. And then you also have some things in the line to even out, because this pump is going to pulse, and you want to get rid of those pulsing. So there's some additional structures like pulse dampers and back pressure regulators and such to smooth out the pumping. Okay. And that is all for today. So we are done. I'm gonna stop sharing. I'll see you on Thursday for the lab on gas chromatography. Have a nice day.